How did life on Earth evolve? The Tree of Life. The Tree of Life represents the relationships between living organisms and their extinct ancestors. Like a family tree, consisting of a great diversity of life forms and including all organisms that live or have lived on our planet, including you. How did this biodiversity come about? The last common ancestor of all living beings inhabited the ocean shortly after the formation of the Earth. Self-replicating biomolecules originated first and gave rise to the first cells. The descendants of those cells are called bacteria and archaea, known as prokaryotes. Prokaryotes are characterized by the fact that their DNA is not encapsulated in a nucleus. From the union of two prokaryotic cells, one an archaea and the other a bacteria capable of breathing oxygen, eukaryotic cells were born, which are more complex and have their DNA encapsulated in a nucleus. Those are the types of cells that make up your body. Some of those cells generated their own food from light or chemical energy through photosynthesis or chemosynthesis. We call these ones autotrophic. Others are dependent on organic matter to live. These ones are called heterotrophic cells. A key evolutionary step was the invention of multicellularity, collectives of cells working together to survive. Multicellular organisms evolved into complex structures with tissues and organs performing different functions, eventually giving rise to most of the biodiversity we know nowadays. Darwin did not represent these evolutionary changes as a line in one direction that necessarily signified progress, but instead as a branched tree. And since then, we have used this metaphor to understand the evolutionary relationships between all organisms, both living and extinct. Later on, Haeckel coined the term phylogeny, meaning the formation of branches within this evolutionary tree, and proposed the first major groupings of organisms. But how do we make sense of these trees? The tree begins in the past, with the common ancestor of all species, and its leaves are those organisms. The point where a branch emerges is called a node, and represents the ancestor species of all the species on that branch. Haeckel's classical point of view proposed three main branches for the eukaryotes. One branch grouped the flowering and non-flowering plants together, including less well-known organisms such as ferns and mosses. Another large branch grouped together very diverse organisms such as protists and fungi, which nowadays we know to be independent branches. And finally the animal branch, including organisms as diverse as starfish, comb jellyfish, crustaceans, mollusks, earthworms, and elephants. Some of these have survived to the present day, but trilobites, dinosaurs, ammonites, and Neanderthals went extinct, and we only know them from their fossils. Branches represent the degree of evolutionary relationship between organisms sharing a recent common ancestor, and tell us when those organisms diverge. The longer they are, the longer ago they diverged from their common ancestor. But how do we uncover those relationships? First, we collect species in nature, whose position on the tree of life we want to understand. For some organisms, morphological characteristics already give us clues about their evolutionary relationships. For example, mammals are defined as organisms with 4 legs and covered in fur, like us, tigers, or elephants. However, for other organisms, their morphological characters are less helpful. In these cases, we use molecular methods. We extract their genetic material, deoxyribonucleic acid, or DNA in particular, sequence it in the laboratory, and these sequences, which define to some extent the organism's characteristics, are explored using bioinformatic analysis. DNA is composed of 4 nucleotides: cytosine, guanine, adenine, and thymine. These are arranged differently in the genes of each organism. Thus, the DNA sequence of a particular butterfly gene will have a unique combination of these nucleotides, which will be different from, for example, the DNA sequence of an algae, or coral, or an earthworm. These sequences will be more similar to each other the closer the organisms are evolutionarily. But the evolutionary history of each gene is very complex, so we need to study many genes, or all the genetic material, called the genome, to understand the evolutionary relationships between organisms. These relationships can sometimes change depending on the gene studied. In this example, for gene A, the sequences of butterflies and algae are more similar to each other than those of corals and earthworms. So we would infer that butterflies and algae are evolutionarily closer, and the same for corals and earthworms. And although they would all share a common ancestor, comparing the B gene sequence, the relationships between the same organisms would be different. And comparing the C gene sequences, the order of the relationships would change yet again. However, if we combine all the genes in the analysis, we would surmise that algae were the first group to diverge, then corals, and finally butterflies and worms, which is what we think has happened with these organisms. Sometimes different genes of the same species tell different stories, because each of them has evolved independently, due, for example, to the different adaptations that the organism has undergone. That is why nowadays we know it is so important to study the complete genomes of organisms. Each organism contains an enormous amount of genetic information, which is impossible to compare and analyse by hand. For example, the DNA of your cells consists of billions of A, C, G and T letters, which form more than 20,000 genes. Therefore, we analyse this information using complex mathematical and evolutionary models. These analyses can take up to a year to complete due to the very large number of sequences involved. The reconstruction of the tree of life is a dynamic and constantly developing area of research, and the position of the branches can change a lot depending on the models and algorithms chosen, and the organisms and species included in the analysis. There are still branches of the tree of life that are difficult to place, and it is possible that their location will change once we have spelt out the genomes of many more species and include biodiversity that is still unknown to us. Thanks to new methodologies for sequencing longer DNA fragments and the discovery of new organisms, we are making great strides towards deciphering the tree of life. In Spain, we have a large scientific community working to understand evolutionary relationships and to advance our understanding of the tree of life and its evolution. The collaborative, multidisciplinary network of excellence, Hub CSIC, was created with the aim of connecting this community through diverse communication, training and collaborative platforms, all with the common goal of responding to a global challenge: to stimulate the generation of ideas and transversal projects so that we can understand the origin of life, its evolution and diversity, and perhaps its synthesis in the laboratory.